Hello, and welcome to another episode of BioChat Collective. I'm your host, Evan Figueroa, along with,
Brandon Budine.
And Katie Durazo.
and today we'll be covering Graves' case study number 3, which is based on his paper along with his collaborators called Genomics of Parallel Experimental Evolution in Drosophila. So, firstly, the main research question of this study is how do different selection regimes affect phenotypic and genomic parallelism in Drosophila melanogaster populations over long evolutionary durations? This question arises from the contrast observed between asexual and sexual populations in terms of genetic adaptation. In asexual organisms like E. coli, adaptation often involves selective sweeps of new mutations, especially in the earliest stages of new evolution. In contrast, in sexual populations like Drosophila melanogaster, adaptation appears to depend more on changes in allele frequencies of standing genetic variation rather than the fixation of new mutations. This study investigates whether this pattern holds over 100 to 1,000 generations of sustained selection and probes the degree of parallelism in phenotypic and genomic changes among populations that share recent versus long sustained selection histories. By examining 30 Drosophila populations that have undergone different selection regimes, this research aims to provide insights into the evolutionary mechanisms driving these changes. And to cover the results, the main research, we have Katie.
Okay, and so the study reveals several important findings about evolutionary processes in Drosophila. First, it demonstrates parallel evolution where independent populations evolve similarly despite starting from different genetic backgrounds when exposed to the same selection pressures. Researchers identified specific genomic signatures with genetic loci consistently changing across populations linked to stress resistance, reproduction, and lifespan traits. This suggests that common genetic pathways are involved in evolutionary responses. Although some genetic changes were shared among the populations, the differences observed highlight the complexity of evolution, indicating that both genetic and environmental factors contribute to adaptive processes. Additionally, the study pinpointed genes related to metabolism, stress response, and reproduction, which were critical for the adaptations, and the findings suggest that the parallel evolution can occur due to shared genetic mechanisms, offering valuable insights into how selective pressures shape genetic variations across populations. And then lastly, the study emphasizes that evolution can follow similar paths in different populations with specific genes and common genetic pathways playing a crucial role in adaptation.
And in this study on the experimental evolution of moderately outbred Drosophila populations led to several key conclusions, such as sexual populations evolving quickly and consistently in response to selection, primarily relying on standing genetic variation rather than the emergence of new beneficial mutations undergoing hard selective sweeps. In contrast to clonal organisms like E. coli, where adaptation is driven by selective sweeps and clonal interference, Drosophila populations show changes in allele frequencies that preserve genetic diversity, avoiding the fixation of single haplotypes. The study observed extensive genomic changes, including shifts in single nucleotide polymorphisms, or SNPs, transposable elements, TEs, structural variants, or SVs, and patterns of linkage disequilibrium, LD, with parallel changes seen across populations exposed to the same recent selection regime. This parallelism at both the phenotypic and genomic levels highlights the role of selection acting on pre-existing genetic variation. Furthermore, the rapid loss of differences between long-established and newly derived populations sharing the same selection regime demonstrates the swift effect of standing genetic variation under selection. These findings challenge traditional views of adaptation in sexual populations, showing that they rely on maintaining stable polymorphisms rather than complete fixation of new genetic variants. Overall, the study emphasized the efficiency of sexual reproduction in facilitating rapid and repeatable evolutionary responses, offering valuable insights into evolutionary mechanisms in both laboratory and broader ecological contexts.
And thank you once again for joining BioChat Collective.